Welcome to DermWire News, powered by DermWire.com. DermWire News for Smart Speaker is made possible with advertising support from Ortho Dermatologics. I'm Stephanie Talea, Senior Managing Editor with Practical Dermatology magazine. The naked eye alone is not enough to accurately diagnose skin cancer, and smartphone apps may also miss melanomas, according to a new review published in the Cochrane Library as part of a special collection of Cochrane's systemic reviews, bringing together a large body of research on the accuracy of tests used to diagnose skin cancer. The suite of 11 reviews was led by Dr. Jacques Dines, a senior researcher at the University of Birmingham in the UK, and supported by the Cochrane Skin Cancer Group and a team of more than 30 researchers and expert advisors. Early and accurate detection of all skin cancer types is essential to manage the disease and to improve survival rates in melanoma, especially given the rate of skin cancer worldwide is rising, said Dr. Dines in a news release. The visual nature of skin cancer means that it can be detected and treated in many different ways and by a number of different types of specialists. Therefore, the aim of these reviews is to provide the world's best evidence for how endemic type of cancer should be identified and treated. Overall, the researchers found that careful consideration should be given of all the technologies that could be used to make sure that skin cancers are not missed, at the same time ensuring that inappropriate referrals for specialist assessment and inappropriate excision of benign skin lesions are kept to a minimum. Key findings from the review include visual inspection using the naked eye alone is not good enough and melanomas may be missed. Smartphone applications used by people with concerns about new or changing moles or other skin lesions have a high chance of missing melanomas. When used by specialists, dermoscopy is better at diagnosing melanoma than visual inspection alone, and may also help in the diagnosis of basal cell carcinomas. Dermoscopy may also help general practitioners correctly identify people with suspicious lesions who need not be seen by a specialist. Dermoscopy is already widely used by dermatologists to diagnose melanoma, but its use in primary care has not been widely evaluated, therefore more specific research is needed. Checklists to help interpret dermoscopy might help improve the accuracy of diagnosis for practitioners with less expertise and training. Teledermatology is likely to be a good way of helping GPs to decide which skin lesions need to be seen by a skin specialist, but future research needs to be better designed. Artificial intelligence techniques such as computer-assisted diagnosis can identify more melanomas than doctors using dermoscopy images. However, CAD systems also produce far more false positive diagnoses than dermoscopy and could lead to considerable increases in unnecessary surgery. Further research is needed on the use of specialist tests such as reflectance confocal microscopy, a non-invasive imaging technique which allows a clinician to do a virtual biopsy of the skin and obtain diagnostic clues while minimizing unnecessary skin biopsies. RCM is not currently widely used in the UK, but the evidence suggests that RCM may be better than dermoscopy for the diagnosis of melanoma and lesions that are difficult to diagnose. And other tests, such as using high-frequency ultrasound, have some promise, particularly for the diagnosis of BCCs, but the evidence base is small and more work is needed. The research team said that future studies evaluating diagnostic skin cancer tests should recruit patients with suspicious skin lesions at the point of the clinical pathway where the test under evaluation will be used in practice. Further research is also needed to evaluate whether checklists to assist diagnosis by visual inspection alone can improve accuracy and to identify how much accuracy varies according to the level of expertise of the clinician carrying out the assessment. Well-designed studies of dermoscopy in primary care are needed, and the best ways of delivering dermoscopy training need to be identified, the review authors state. The reviews have been shared with the National Institute for Health and Care Excellence to inform potential update of the NICE melanoma guideline, which was last updated in 2015. The FDA has granted fast-track designation to Asano Biosciences investigational oral JAK and SYK dual inhibitor, ASN002, for the treatment of moderate to severe atopic dermatitis. We are pleased that the FDA has granted fast-track designation to ASN002. The designation recognizes the importance of accelerating the development of new medicines for the treatment of challenging dermatological inflammatory diseases that have a major impact on patients' daily quality of life, says Sandeep Gupta, founder and CEO of Asana. We look forward to taking advantage of the opportunity for frequent interactions with the FDA throughout the development of ASN 002 and the potential expedited review offered by their Fast Track program. ASN 002 is currently being evaluated in moderate to severe atopic dermatitis in the Phase 2b RADIANT study. It is also being evaluated in a Phase 2 trial in patients with severe chronic hand eczema. ASN 002 is the first oral drug to demonstrate improvement in atopic dermatitis lesional skin phenotype correlating with clinical efficacy. ASN 002 data demonstrating improvements in skin pathology, disease-related genes, and inflammation biomarkers correlating with clinical efficacy in patients with moderate to severe atopic dermatitis will be presented at the Inflammatory Skin Disease Summit meeting to be held in Vienna, Austria on December 12th through the 15th. Thank you for listening to DermWire News, powered by DermWire.com. This editorially independent program is supported with advertising from Ortho Dermatologics.